Hold your hand out in front of you and look at two fingers in particular, the index finger and the ring finger. For most people, one of these fingers is very slightly longer than the other, sometimes by a fraction of a millimeter, sometimes by several. It seems like a trivial detail, the kind of thing nobody would think to measure, and yet this exact comparison, known in scientific literature as the second to fourth digit ratio, or simply 2D to 4D, has become one of the most studied and most argued about measurements in human biology, generating hundreds of published research papers over the past few decades, and more recently, a genuine genetic explanation for where the difference actually comes from. The measurement itself is almost embarrassingly simple. Researchers take the length of the second digit, the index finger, and divide it by the length of the fourth digit, the ring finger, both measured on the same hand, from the crease at the base of the finger to its very tip. If the index finger is shorter than the ring finger, the resulting ratio comes out below 1, and scientists describe this as a low-digit ratio. If the index finger is longer than the ring finger, the ratio comes out above 1, and this is described as a high-digit ratio. This simple number was first documented as far back as 1875 by a researcher named Ecker, who noticed the pattern but had no explanation for it and no tools capable of investigating its cause. The measurement was then largely forgotten for more than a century until it was rediscovered and popularized in the early 1980s by researchers examining patterns of sexual dimorphism in the human skeleton, and it has since become a small but genuinely active subfield of biological research, particularly within the study of prenatal hormone exposure. The reason scientists became so interested in something as seemingly minor as relative finger length comes down to embryology. During the first trimester of pregnancy, at a developmental window that researchers have pinned down to roughly the 14th week of fetal growth, the same genetic instructions that guide the formation of a fetus's reproductive organs are also, at the very same time and through many of the very same genetic pathways, guiding the formation of its fingers and toes. The genes responsible for this shared developmental process belong to a family known as the HOX genes, specifically the HOXA and HOXD clusters, a set of genetic instructions that acts almost like a blueprint controlling how different body segments are laid out during early development. Because these same HOX genes influence both the differentiation of the gonads and the relative growth of the digits on the hand, researchers began to suspect and eventually to demonstrate through a range of indirect evidence that the hormonal environment surrounding a developing fetus, specifically the relative balance between testosterone and estrogen circulating in the womb during this critical early window, leaves a lasting physical signature not just in the reproductive system, but in the proportions of the hand itself. The working theory, generally called the organizational hypothesis, holds that higher relative exposure to testosterone during this specific prenatal window tends to produce a longer ring finger relative to the index finger, resulting in a lower digit ratio, while higher relative exposure to estrogen tends to produce the opposite pattern, a longer index finger relative to the ring finger, and therefore a higher digit ratio. Because male fetuses are, on average, exposed to substantially more testosterone during early development than female fetuses, this theory predicts that men should show lower average digit ratios than women. And this is, in fact, one of the most consistently replicated findings in the entire body of digit ratio research. Across dozens of independent studies conducted in different countries, different ethnic populations, and different research laboratories, men reliably show a lower second to fourth digit ratio than women on average. The sex difference appears to establish itself very early in development and remains relatively stable across a person's lifetime, meaning that an adult's digit ratio today is generally a reasonably faithful echo of the hormonal environment that surrounded them in the womb decades earlier, at least according to the researchers who have spent their careers studying this measurement. Interestingly, this sexually dimorphic pattern in relative digit length is not unique to humans. Researchers studying other mammal species, including mice, have found comparable differences in the relative length of digits between males and females, and in some of these animal models, scientists have been able to go a step further than is possible in human research, directly manipulating hormone exposure in developing embryos and observing the resulting change in digit proportions. These animal studies have generally supported the underlying hormonal theory quite strongly, since researchers can control the experimental conditions in ways that would obviously be impossible and unethical in human pregnancy. And this cross-species consistency is one of the reasons the organizational hypothesis has remained the leading explanation for digit ratio for as long as it has, even as the human behavioral research built on top of it has run into much rockier territory. Some of the strongest supporting evidence for this hormonal explanation in humans comes not from healthy population studies, but from clinical research involving a rare condition known as congenital adrenal hyperplasia. a disorder in which the fetal adrenal glands are enlarged and produce unusually high levels of androgen hormones during development. Children born with this condition, when compared to typically developing children of the same sex, consistently show lower, more masculinized digit ratios, a finding that has been confirmed across multiple studies and even summarized in formal meta-analyses pooling data from many separate research groups. This clinical evidence matters because it provides something population studies alone cannot, a scenario in which researchers already know, independently of the finger measurement itself, that a group of individuals experienced unusually high prenatal androgen exposure, and then can check whether their digit ratios actually reflect that known biological history. Twin studies have added another layer of support to the underlying biological picture by comparing digit ratio similarity between identical twins who share essentially all of their genetic material and fraternal twins who share on average only about half. Researchers have been able to estimate how much of the variation in digit ratio across a population is attributable to genetics at all, as opposed to environmental or purely random developmental factors. These twin study estimates have generally landed in a similar range to what later genetic studies would go on to confirm directly, giving researchers a reasonably early, if indirect, signal that digit ratio was a substantially heritable trait long before anyone had identified a single specific gene responsible for it. For years, this left an obvious and frustrating gap in the science. Researchers had a reasonably solid hormonal theory, decent clinical support from conditions like congenital adrenal hyperplasia, supportive animal research, and a robust, repeatedly confirmed sex difference between men and women. What they did not have, for a very long time, was a clear picture of the actual genes responsible for producing variation in digit ratio among individuals of the same sex, the kind of hard genetic evidence that would let scientists move beyond correlation and hormonal inference toward an actual molecular explanation. The largest and most rigorous genetic investigation of digit ratio to date pulled together genetic data from more than 15,000 people in its primary discovery sample, then tested its findings again in a much larger replication sample of more than 75,000 additional people, a scale of investigation that dwarfs almost every previous study on this topic, most of which had worked with sample sizes in the dozens or low hundreds. This large scale genetic analysis identified 11 distinct locations in the human genome that showed a reliable statistical association with digit ratio, 9 of which had never been previously linked to this trait in any prior research. Among the genes implicated in this research and in related molecular studies of limb development are SMOC1 and Lin28B, both of which play well-established roles in the very early stages of limb formation in the developing embryo, precisely the kind of genes you would expect to be involved if digit length truly is shaped by processes occurring deep in the earliest weeks of prenatal development, rather than by anything happening later in childhood or adolescence. Lin28B, in particular, has also been separately implicated in research on the timing of puberty and overall body growth, which has led some researchers to speculate that digit ratio may be entangled with a broader shared genetic network governing growth timing. Generally, rather than being controlled by a pathway that is entirely specific to hand development, though this remains an area of ongoing investigation rather than settled fact. The genetic study also looked specifically at a different genetic marker entirely, a repeating sequence of genetic code inside the androgen receptor gene itself, sometimes described as a CAG repeat, which is already known from separate research to influence how sensitive a person's cells are to testosterone once it is present in the body. Instead, digit ratio appears to emerge from the combined, overlapping influence of multiple genetic variants, each contributing a small effect layered on top of the broader hormonal environment established in the womb, with researchers estimating overall that somewhere in the range of 60 to 70% of the variation in digit ratio between individuals can be attributed to genetic factors, leaving a meaningful remaining share influenced by other prenatal and possibly early developmental conditions that are much harder to measure directly. This is genuinely important context because it means the popular idea of digit ratio as some kind of simple one-to-one readout of your personal testosterone exposure is an oversimplification of what the actual data shows. The genetic and hormonal evidence supports the idea that digit ratio is a real, biologically meaningful, partly heritable trait connected to prenatal hormone exposure. It does not support the idea that measuring someone's fingers gives you a precise, individualized hormone reading. And this distinction matters enormously once you start looking at the huge and controversial body of research claiming that digit ratio predicts all sorts of other things about a person. Part of the difficulty in this field has always come down to something as basic as measurement method itself. Early digit ratio research often relied on simple handheld rulers or calipers applied directly to a person's hand, a method that introduces real measurement error, particularly across the small fractions of a millimeter that often separate one person's ratio from another's, Later researchers turned to photocopying or scanning the hand and measuring the resulting image, which improved consistency somewhat, while the most rigorous studies have used radiographic imaging, essentially X-rays of the hand, to measure the bones themselves rather than the soft tissue of the finger, removing the influence of things like finger fat, swelling, or skin thickness from the measurement entirely. Over roughly the past 2 decades, researchers have published well over 300 papers attempting to link digit ratio to an extraordinarily wide range of human traits, behaviors, and health conditions. Some of the more commonly cited claims connect a lower digit ratio, the more masculinized pattern associated with higher prenatal testosterone, to traits such as increased physical aggression, greater risk-taking behavior, higher athletic performance, particularly in sports requiring speed and power, increased sperm count and fertility markers in men, and higher scores on self. Reported measures of general competitiveness. Several sports science studies have specifically compared digit ratios among elite athletes, including runners, footballers, and other competitors in speed and endurance-based sports, against those of the general population, generally reporting that elite athletes in these disciplines tend to show lower average digit ratios than non-athletes, though critics of this research point out that many of these athlete studies involve fairly small samples of elite competitors and do not always control adequately for other factors, such as body size or training history, that could plausibly explain at least part of any observed difference. The honest scientific picture here is considerably messier and more cautious than most popular articles about digit ratio tend to suggest. A significant portion of this research has involved fairly small sample sizes, often just a few hundred participants or fewer, and a persistent problem across the field has been an inability to reliably reproduce many of these findings when other research teams attempt to repeat the same study with a new, independent group of participants. One well-designed study that specifically set out to test whether digit ratio predicts competitiveness looked at 2 separate and reasonably large samples, one drawn from shopping mall visitors and another from university students, and measured competitiveness in 2 different ways, once through actual behavior in structured economic experiments, and once through simple, self-reported questionnaires. This kind of split result, where a trait correlates with what people say about themselves, but not with what they actually do when placed in a real decision-making scenario, is exactly the sort of finding that makes careful researchers cautious about overselling digit ratio as a meaningful behavioral predictor, since self-reported personality questionnaires are subject to all sorts of influences that have nothing to do with prenatal hormones. A separate line of research, looking specifically at women, found an even more direct challenge to the popular claims surrounding digit ratio. Using a precise radiographic method of measuring finger length, considerably more accurate than the simple ruler and caliper measurements used in many earlier studies, researchers examined a large sample of women and looked for any relationship between digit ratio and a whole battery of anthropometric, behavioral, nutritional, health-related, occupational, and fertility-related variables. They found essentially no meaningful relationship across the board, a genuinely important null result in a research field that has often been criticized for publishing positive, attention-grabbing findings while quietly failing to publish the far more common negative results that do not confirm a hypothesized link. This pattern, sometimes called publication bias, is a broader problem across many areas of behavioral science, but digit ratio research has been specifically flagged by methodologists as a field where it appears to be especially pronounced, given how many small studies reporting a striking positive finding exist for every large, well-controlled study that quietly reports finding nothing at all. Findings like this have led a meaningful portion of the scientific community studying digit ratio to argue that the field, taken as a whole, needs to be much more careful about distinguishing between genuinely robust findings, such as the basic sex difference between men and women, and the connection to clinical conditions like congenital adrenal hyperplasia, and the much shakier, frequently unreplicated claims about personality traits, athletic ability, or disease risk that tend to dominate popular coverage of the topic. It is also worth being clear that digit ratio itself is not a fixed, universal number even before you get into questions of what it might predict. Average digit ratios differ measurably across different ethnic populations, even though the basic pattern of men showing lower ratios than women within any given population remains remarkably consistent across every ethnic group researchers have studied, which is itself an interesting finding since it suggests that whatever biological mechanism produces the male-female difference is a deeply conserved feature of human development, even as the overall baseline ratio shifts somewhat from population to population for reasons that are not yet fully understood, and that likely involves some combination of genetic ancestry and possibly broader environmental or nutritional factors during pregnancy that researchers have not yet fully mapped out. The popularity of digit ratio as a topic of public fascination has also fed into the growing direct-to-consumer genetic testing industry. Several companies that analyze raw genetic data uploaded by customers now offer a digit ratio prediction as one of dozens of lifestyle and physical trait reports, included alongside more established genetic findings like ancestry composition. And these companies are generally careful in their own fine print to note that such predictions reflect research, level genetic associations rather than a diagnostic or deterministic reading of a customer's actual measured finger length, and that factors such as which hand is measured, the specific measurement method used, age, and broader population background can all affect how closely a person's actual physically measured ratio matches what their genetic data alone would predict. More recent research has continued to push this field in new directions, rather than simply rehashing old claims about personality and athletic performance. Within just the past couple of years, researchers working out of Swansea University's Applied Sports, Technology and Medicine Research Group, collaborating with anthropologists at Istanbul University, have been investigating a genuinely different angle on digit ratio, exploring what the same prenatal hormonal balance that shapes finger length might also reveal about patterns connected to human brain evolution, building on the underlying idea that the same early developmental hormonal environment influencing limb growth may leave parallel traces in aspects of brain and skull development that differ between men and women and across human populations more broadly. This kind of research represents a meaningful evolution in how scientists are approaching digit ratio, moving away from the simpler and often overhyped personality and behavior correlations that dominated the field for its first couple of decades, and toward more careful, mechanistically grounded questions about shared prenatal developmental pathways, using digit ratio as one small measurable window into a much larger and more complex developmental story. Taken together, what the genetics and the broader research record actually tell us is a story that is more interesting and considerably more honest than the simple version that tends to circulate online. Yes, the length of your ring finger relative to your index finger really does appear to be shaped, at least in part, by the balance of testosterone and estrogen. Your body was exposed to in the womb during a specific early window of fetal development, and this connection is now backed by real, large-scale genetic evidence identifying actual genes involved in the process, genes that also play a broader role in early limb formation. Yes, this produces a real, measurable, and remarkably consistent difference between men and women that shows up across virtually every human population studied. And yes, the same basic pattern of sexual dimorphism appears in other mammal species as well, giving the underlying hormonal explanation a genuinely solid evolutionary and comparative foundation. And yes, clinical evidence from conditions involving abnormal prenatal hormone exposure provides genuine independent support for the underlying biological mechanism. But no, this does not mean that measuring your own fingers right now will reliably tell you anything precise or individually diagnostic about your personality, your athletic potential, your fertility, your risk of disease, or your position on any behavioral spectrum, however popular and appealing that idea might be. The genetic variants identified so far explain only a small fraction of the overall variation in digit ratio across people. The trait itself is influenced by a tangle of genetic and non-genetic prenatal factors that are still not fully mapped out. Measurement technique itself introduces more inconsistency into this field than many popular accounts acknowledge, and the vast majority of headline-grabbing claims connecting digit ratio to specific personality traits or life outcomes have either failed to replicate reliably across independent studies or have simply never been tested with the kind of large, carefully controlled sample size that would be needed to trust the result. The honest scientific position, reflected in the more rigorous and more recent research, is that digit ratio is a genuinely fascinating window into a very specific slice of early human development, a small, permanent physical trace left behind by a hormonal moment that occurred before you were even born. But it is not a fortune-telling device, and no single measurement taken from the back of your hand is ever going to substitute for the far more complicated genetic and environmental story that actually shapes who a person becomes.